Right now, as you watch this, your cells are running the largest coordinated strike operation in human biology. Sirtuins are the stealth bombers. NAD+ is the fuel in the tank. NMN is the supply line keeping the whole fleet airborne. And somewhere in your liver, a senescent zombie cell is about to get a 30,000-pound molecular bunker buster dropped directly onto its corrupted DNA. Here's the play-by-play of the operation your body runs every single day, without asking Congress for approval. Let's talk about the enemy. Every day, your body produces roughly 10,000 precancerous mutations. That's not a typo, 10,000. If your cellular defense forces took the weekend off, you'd be a tumor by Tuesday. The primary target tonight? Cellular senescence. These are zombie cells, technically alive, metabolically active, but refusing to die. They sit there pumping out inflammatory signals like a jammed radio transmitter, corrupting the cells around them. And cancer? Cancer is what happens when one of these corrupted cells figures out how to flip the kill switch off permanently. But here's the thing. Your body does not care about your feelings. Your body has been running counterinsurgency operations since before humans walked upright. And the main battle group? The sirtuin family of proteins. 7 of them. Sirt1 through Sirt7. Think of them as 7 different aircraft in the same strike package, each with a different payload, each with a different mission profile, but all flying toward the same target. Sirt6 is the B-2 Spirit of your cellular defense fleet. It flies dark, it flies deep, and when it locks onto a target, a cancer cell with corrupted DNA, it doesn't just damage it, it doesn't wound it. It deacetylates the histones wrapped around that corrupted DNA, essentially ripping the protective scaffolding off the enemy's command center. Without that scaffolding, the cancer cell can't read its own replication instructions. It's not dead yet, but it's mission killed. The genes that tell it to keep dividing? Silent. The genes that tell it to self-destruct? Activated. But here's where it gets sporty. Sirt6 doesn't work alone. Just like the B-2 needs its tankers, its F-22 escort, its satellite guidance, Sirt6 runs on a fuel called NADPlus. Nicotinamide adenine dinucleotide, NADPlus, is the JP-8 jet fuel of every sirtuin operation. Without it, Sirt6 is a $2 billion bomber parked on the runway with empty tanks. Here's the brutal math. By age 50, your NAD+ levels have dropped roughly 50% from your 20s. That means half the bombers are grounded, half the missions don't fly, half the rogue cells get away. This is the part of the briefing where someone usually says, well, can't we just refuel? And the answer is yes. NMN, nicotinamide mononucleotide, is the KC-135 tanker flying mid-Atlantic in a thunderstorm, threading the refueling boom into NAD+ production pathways at 400 knots in pitch darkness. Without NMN, NAD+ production sputters. With NMN, you're topping off the tanks while your sirtuins stay on station. But before Sirt6 can drop its payload, the corridor has to be sanitized. Enter Sirt1 and Sirt3, your F-22 Raptors. Sirt1 runs the high-altitude sweep, deacetylating P53, the protein that decides whether a damaged cell lives or dies. Sirt3 works the mitochondrial deck, keeping the energy factories humming so the whole strike package doesn't lose power mid-mission. Together, they create a 50-mile-wide corridor through your inflammatory landscape where your sirtuin forces operate with total impunity. Not a single inflammatory cytokine gets through. Not one corrupted cell escapes surveillance. This is air superiority at the molecular level. At exactly T-0, the bomb bay doors open. But the weapon isn't a GBU-57 massive ordnance penetrator. It's an autophagosome, a double-membrane vesicle that forms around the target cell like a 30,000-pound hardened steel casing. The process is called autophagy. Your cells literally eat themselves. Not in a panic, not randomly, with GPS-guided precision. The autophagosome forms. It engulfs the zombie cell. It fuses with a lysosome, basically a bag of acid and digestive enzymes, and the enemy is dissolved into raw materials your body recycles to build new, healthy cells. That's the part nobody talks about. Your body doesn't just delete the enemy. It takes the enemy apart, sorts the pieces, and uses them to build reinforcements. It's the most efficient military logistics operation ever designed, and it runs silently, in the background, every single day. So what happens when it fails? When NADPlus drops, when sirtuins are silenced, when autophagy slows to a crawl. The zombie cells pile up. The inflammatory signals multiply. The surveillance grid goes dark. And somewhere in that chaos, one corrupted cell gets lucky. It bypasses the kill switches. It starts replicating. And 18 months later, you're sitting in a doctor's office hearing a word that starts with C. This isn't speculation, it's the mechanism. And understanding the mechanism is the difference between being a passenger in your own biology and being in the cockpit. So what can we actually do about it? 3 things. Number 1, NADPlus restoration. NMN supplementation is the most direct route. Clinical trials show it raises blood NADPlus levels within 30 minutes of oral intake. The dosing window appears to matter. Morning administration aligns with your circadian peak of NADPlus production, and pairing it with exercise amplifies the effect because muscle contraction itself triggers NADPlus synthesis. Number 2, polyphenols. Compounds like resveratrol and pterostilbene don't just activate SIRT1. They dock into a specific binding pocket that researchers at Harvard identified, essentially flipping the enzyme from standby mode to active duty. Number 3, heat shock. Sauna use at 80 degrees Celsius for 20 minutes triggers a cascade that boosts sirtuin expression by up to 40%. Your cells interpret the heat as a threat, and they mobilize the entire defense fleet in response. Enter Spermidine. If NMN is the fuel tanker and sirtuins are the bombers, Spermidine is the munitions factory. This polyamine compound, first isolated from human semen, is one of the most potent natural autophagy inducers ever studied. It works by inhibiting a specific acetyltransferase called EP300, which normally puts the brakes on autophagy. Spermidine cuts that brake line. The result? Your cellular cleanup crews go from maintenance mode to full-scale offensive operations. The number 1 whole food source? Wheat germ. A single tablespoon delivers roughly 6 milligrams, within the range used in the landmark human trials showing cardiovascular and cognitive benefits. It costs pennies. It's sitting on health food store shelves right now, and it's one of the most under-discussed molecules in the entire longevity field. Here's the takeaway. Your body runs Operation Cellular Strike every single day. Not because you remembered to take a supplement, not because you exercised this morning, but because 3.7 billion years of evolution built an autonomous defense network into every one of your 37 trillion cells. Sirtuins, NADPlus, NMN, Spermidine. These aren't separate interventions. They're interdependent components of a single integrated strike system. And the message from the latest research is unmistakable. We are no longer victims of our biological clocks. Your cells are listening. Give them the signal to stay young, stay vibrant, stay curious, and most importantly, stay activated.